Okay, we are ready to get started. Hi, everyone. I'm Rosie Bayari, and I'm part of the software product design team at Element Biosciences, working as a product owner with our software suite, system instruments, some bioinformatics, some cloud work. And I'm really excited to welcome everyone today to our webinar from our webinar series. And with today's topic, AVITI Analysis at Any Scale, Flexible, Powerful, and Secure Analysis with Seqera. I'll start off with a few brief announcements before we begin our talks, and then we'll be able to get started and dive into this great content. So first of all, we have a new program, the AVITI Accelerator Grant Program, AVITI for All. It is open for proposal submissions. Tell us how up to 32 library preps and 2 flow cells of AVITI sequencing would accelerate your research and have a chance to win. The details for this are all on our elementbiosciences.com homepage, so go there, first splash, check it out. Next, the presentation portion of this webinar will be followed by a questions and answer session. Please submit your questions throughout the talk at any point by clicking the Q&A icon at the bottom in your Zoom webinar. There's a chat icon as well. They look very similar, so if possible, use the Q&A icon to submit those questions. Lastly, we are recording this webinar. It's going to be available for viewing in the next couple of days. So if you want to listen again or you're joining remotely after, take a look for that on our socials when we release that. Okay, so I would like to introduce our first speaker, Dr. Harshal Patel. Dr. Patel holds a PhD in bioinformatics from Oxford Brookes University, UK, and he worked as a senior bioinformatician at the Francis Crick Institute in Cancer Research UK before joining Seqera as head of scientific development. He's got over 10 years experience in the field of genomics and has co-authored over 40 peer-reviewed publications. And at Seqera, he's working on helping leading a team of bioinformaticians to streamline usage of Nextflow for pipelines, and using all of that on the Seqera platform. And he's very excited about bioinformatics in general and Nextflow and powerful tools. So let's welcome Dr. Patel and learn a bit about the Seqera platform.
Awesome. Thank you, Rosie. And thank you for inviting me to give this talk as part of your webinar series. Okay, so what I wanted to talk to you about is Seqera and the Seqera platform, a bit about Nextflow as well, which some of you may have heard of, and really what we're trying to frame Seqera as a company that really is trying to develop the modern biotech stack and enable scientists of all levels of experience to essentially be able to analyze their data. So Seqera started out about 5 years ago now. We recently had an anniversary. Nextflow itself, which is a language with which you can write pipelines. Nextflow has been used quite heavily, mostly in genomics, but it's now fanning out to other disciplines where essentially you may want to analyze a piece of data and you need to write some sort of pipeline, which is a series of steps to analyze that data. And Nextflow has really changed the game on that front with some of its core features. And so Nextflow actually has been around for 10 years now almost. It started out in a lab at the CRG, and Evan and Paolo, who are the CEO and CTO of Seqera, were heavily involved in the project, and you know we maintain and we create Nextflow still now at Seqera to push its development and all the benefits that are now being sort of realized across the world. So Seqera was founded, as I said, 5 years ago, and we are doing various things at Seqera. We are trying to essentially make it a lot easier for scientists to analyze their data using Nextflow as the engine, essentially. We have a massive commitment to open source. So Nextflow itself started as an open source project. It's going to remain an open source project. It's very important to us and our leadership that we are committed to open source. In the same sort of vein, Some of you may have heard of NF Core, and so NF Core actually started quite early on in the development of Nextflow, where a community of people came together to try and develop a set of core pipelines that could be contributed to globally, rather than each institute having their own version of a pipeline with its own niceties and hard-coded paths and various other things that make it impossible to run on different infrastructures. NF Core really came together to exploit the benefits that Nextflow offers in terms of creating this community that we can use to develop a core set of pipelines in an open way and set some sensible guidelines in terms of how these pipelines are developed, so they just get better and better. So this is an example of a pipeline. I'm sure a lot of you have heard of RNA-seq data. And so one of the gold standard pipelines on NF Core is the RNA-seq one. That's mostly because there's just tons of RNA-seq data knocking about. But also it's also tended to be the one where we push our most cutting edge changes in terms of Nextflow development and iterations of how NF Core adopts Nextflow within the community. It's generally, again, via consensus decisions across the community to iterate on making these pipelines better, not only from a functional perspective, like having different routes and analysis processes in the pipeline, but also in terms of how best to write Nextflow pipelines. So each pipeline is a series of steps, as you can see here in this tube map. Each step is then chained together in some sort of way, and you start with some sort of raw data, which could be often a AVITI instrument, for example, a FASTQ file, and then you would then take that through a series of steps to generate some sort of result at the end of this pipeline that you can then pass on to scientists to then interrogate the results, whether that's differential expression analysis or looking at UMAPs or various other things. The idea behind these pipelines is to really streamline and make it easy for you to go from raw data to something that a scientist can pick up and work with downstream. And of course, it doesn't end there, because a list of differential genes is then needs to be plugged into a differential analysis software like DESeq, which then might need to be explored in an interactive notebook to make sense of your differential genes and so on. And so it's a series of steps that really you can see that the whole sort of data analysis lifecycle. And Nextflow really, as I said, really changed the game in terms of the way that you write these pipelines. There are a number of benefits that you get from using Nextflow. First of all, it's a DSL. It's built on top of Groovy, which itself is built on top of Java, and so you can inherit the functionality from Groovy, which is actually quite groovy. Pardon the pun. So what you can do with Nextflow is you can link in your existing programming languages, whether that's R, Python, Perl, Fortran, if you've been around for a long time. And you can write those and put those in a bin directory in your Nextflow pipeline, and these will automatically be invoked as part of your pipeline execution when you stitch them together as processes as part of your pipeline. And so you don't really need to change much. You can adopt and change your existing pipelines as much as you want, really. You have the flexibility of doing things from scratch or integrating your existing processes. Another massive advantage is containerization. Before Nextflow, as I mentioned before, for example, when I was at the CRIC, I used to use what's called environment modules on my HPC. And that would be where our HPC admins would install the software locally on our cluster, which then itself has a bunch of dependencies, which then have a bunch of dependencies, and is very tightly knit to that particular environment. And so if I try and share my pipeline with a collaborator or someone else that wants to reproduce a result that I've just generated, it's virtually impossible, because I'd have to physically go into the basement, pick up a HPC and give it to my collaborator in order to replicate that result. Now, with containerization and things like Docker and Singularity, you're essentially running each of those isolated tasks within containers. And so if you're running BWA or SAM tools or whatever it is, these containers come pre-built with these tools already there. And so in terms of sharing this software wherever you want, it's made it stupendously easy to do that. And as I said, it's really changed the game in terms of how portable these pipelines have become. Another big factor is GitHub and version control. With these repositories, you can essentially collaborate on how you develop your code. Everything is version controlled. You know exactly who's done what. You can have review processes in place to improve your pipelines and CI/CD type testing to ensure that they're working. And again, Nextflow really sort of changed the game this way in terms of how people collaborate on writing these pipelines. But on top of all of that, these pipelines can run virtually anywhere. If you write them in the correct way with minimal assumptions about where they're supposed to be run, you can hook in configuration on top of the pipeline to suggest that, okay, today I've woken up and I feel like running this pipeline on AWS. Tomorrow I want to run it on GCP. The day after I'd like to run it on Azure. And so Nextflow really gives you the flexibility of running these pipelines wherever you want. You write a pipeline once and you can run it virtually anywhere. The community itself for Nextflow has grown almost exponentially over the years, and it's just going from strength to strength. We're seeing more and more adoption of Nextflow, as I said, in various different disciplines. Anything you can containerize as a pipeline, you can write a Nextflow pipeline for. So anything you can run in containers, you can use use with Nextflow, and so that illustrates it doesn't necessarily just have to be for genomics, proteomics, metabolomics. We have people on NF Core coming to us to add earth sciences pipelines and various other types of pipelines too. I briefly gave you an overview of what NF Core is. It's a community which kind of sits alongside the Nextflow community. Both of these have their own communication channels, which are awesome. If you want help with Nextflow or if you want help with NF Core, I definitely recommend joining those communication channels, which you can access via the website. You can just Google them and find them there. And there are a number of benefits, again, for having communities like this, because me as an expert potentially in genomics, I'm not an expert in metabolomics or proteomics or protein folding. Whereas having an open source community like this means that people with the right expertise and domain knowledge can come in and really contribute pipelines to a core set of contributions, which then becomes useful for everyone else. And so on the left, you can see a word cloud of some of the more popular pipelines that we have. And you've got the green ones in genomics, you've got red ones, which are proteomics, blue ones, metagenomics. You've got an image analysis pipeline. Again, image analysis adoption on Nextflow is growing a lot because some of these images and data you're getting off these instruments are huge, and it's impossible to open them up on a desktop. You need to try and batch process as much of this as you can via traditional compute methods or in the cloud. So Nextflow is open source, and as I said, it will stay open source. NF Core is open source. It will stay open source. A lot of the key contributors to NF Core are actually at Seqera, and as I mentioned, that ties into our commitment to open source. We're still pushing the limits and boundaries of what we can do and contribute to these open source layers. But we also have a licensed product, which is called Seqera Platform. And Seqera Platform it was previously called Tower, for those of you that may have referred to it as that, just to avoid any confusion. It's still very sort of hot off the press, so I won't excuse you for making any mistakes. I've made tons already, and it's been a couple of weeks. So what Seqera Platform does is it's essentially a layer on top of Nextflow that allows you to do a ton more things than you would just by running Nextflow in isolation. So Nextflow is a CLI tool. It's typically run in isolation it's typically run in isolation by users on a command line type interface. But then that's not intuitive for users that may not want to use a command line or can't use a command line and want something a bit more easy to manage. But on top of that, there are tons of other features that we've added to the platform that really take the adoption of Nextflow to the next level, like tracking cloud expenses and reducing those, making it easier to maintain and manage teams and infrastructure and various other things via this platform in a unified type of way. You can spin up a compute environment very quickly to run your pipelines in. You can have users have access to that very quickly. And it just allows you to collaborate and maintain and manage the way that you're running Nextflow within your own organization. And so, as I mentioned earlier briefly, that data analysis doesn't really stop just at a Nextflow pipeline running. You run an RNA-seq pipeline, you start with FASTQ files, you end up with some sort of differential counts in the case of NF4 RNA-seq. But then you want to take those differential counts and you want to do some statistical analysis on those. And then you might want to take the results of that statistical analysis and load up an interactive notebook. And then you might also, throughout that process, want to share those results with collaborators. And what we're really trying to do as part of our roadmap is to fill that whole end-to-end cycle where you start from some raw data and you end up being able to collaborate with your colleagues to actually come up with some results that you can eventually, hopefully, put in a scientific publication. Parts of this are stuff that we're working on. We're also filling out a ton of features as well recently that have been announced at the summit in Barcelona that we hosted last week, which is one of our flagship events. We've also got a summit coming up in Boston in a few weeks, and I'll mention that right at the end. We have customers of all shapes and sizes: big pharma, CDC, Johnson & Johnson, Moderna, small biotechs, as well as academic institutions like the Sanger and so on, that are, have vested a lot of their time and effort into really standardizing the way that they analyze their data using Nextflow and the Seqera platform. We also have vendors like Element as well as customers who, who's a pleasure to work with, and also Element themselves are contributing actively to NF Core through the demultiplexing pipeline, which is more relevant for sequencing instruments and so on, and really sort of buying into what we're trying to do at NF Core, which is refreshing and is also to some extent changing the way that open source is visualized and viewed, not only from a vendor perspective, but also from a pharma private industry type perspective. Okay, so as a result of our recent rebrand on Seqera platform, we have a core set of products. One of those is Nextflow open source. Another is MultiQC. Phil Ewells, who's been working on that through blood, sweat, and tears for years. It's a very commonly used bioinformatics tool in a lot of pipelines that are out there. And I think Brian might show you some of that a bit later on. So he's on staff as well. That will remain open source. We're able to recruit as part of taking this on within Seqera. We're able to recruit engineers to work on this actively to push that agenda forward. We've recently announced we're open sourcing Wave, which is another product that we've developed that really makes it a lot easier to interact and build and provision containers via your Nextflow pipelines, and that will be made open source as well. Fusion is this tech stack essentially that we've built up that allows you to really change the way that you interact, especially in cloud environments. And so typically in cloud environments, when you run a pipeline, you are using blob storage, which is different from your typical HPC type storage. So with blob storage, what you have to do is you have to copy data from one bucket into the virtual machine where you're running your Nextflow task for a given tool, whether it's BWA or FastQC, process that data, and then publish the data out into, say, another bucket location or a similar bucket location. That can be quite wasteful and it's fragile because you're at the mercy of the cloud APIs to copy tons of data around, which can be quite huge for genomics pipelines, especially. And you start thinking about protein folding pipelines or image analysis where it's even bigger. This causes bottlenecks and issues where you physically takes ages just to copy the data into the task to run anything sensible. Whereas with Fusion, what we've done over the past year and a half is developed and matured it in a way where it acts like you mounting your data within the task. So rather than copying it in, you're mounting that bucket directly into the container. And there's some clever things that go on behind the scenes. If you use Fusion in conjunction with these NVMe disks in the cloud, it really speeds up IO operations and the way that you can process this data in bulk, especially for large data. And so it's now relatively stable, and we will be really looking to push it out soon. And there are lots of benefits that you can have, as I mentioned, in terms of making your pipelines a lot more efficient just by using something like this, in terms of the time it takes to run your pipeline, but also how much it actually then ends up costing you in the cloud. So briefly for each of these, as I mentioned, Wave allows you to provision containers. It's got some really neat things behind the scenes that it does to really make it a lot more easier for you to develop your pipelines and also then potentially inject binaries and various other things within containers on the fly for you to provision these containers when you're running your pipeline. Fusion, as I alluded to, the costs and the benefits you can get from this are kind of highlighted in this plot here, where you can see Fusion is almost twice as fast than standard S3, and it's on par or even slightly better than using FSX, which is a hosted a file system type solution that is available on AWS, but is a lot more expensive than using plain S3. And of course, Fusion uses plain S3 behind the scenes, and so you mitigate for those costs directly there. MultiQC, this is an example report of what MultiQC generates, and it's used a lot, as I said, in the bioinformatics space to aggregate results across experiments to summarize them nicely. More recently, at the summit in Barcelona, we announced the Data Explorer functionality that we're bringing out, and this is essentially a way of you using your existing credentials in the platform to be able to browse buckets, whether that's public buckets like the 1,000 genomes or the SRA, or your own internal buckets. You can browse them directly via the platform. And so this just means that whenever you're running a pipeline, maybe you want to troubleshoot and your pipeline breaks for some reason, and you want to go straight to the work directory of a task, we can now link directly to that bucket and that particular directory via the platform itself, rather than you having to go outside and find this data via a console. And so we've added upload, download functionality. It's now in public preview on our cloud platform. And it's really going to allow users to have and access their data via one unified interface, a bit like a file browser for cloud, essentially, within the platform. We've also brought out and announced the Data Studios functionality at the summit. And this is really where that downstream analysis ties in that I've mentioned, where you can take some sort of differential list of genes and use more interactive environments like Jupyter or RStudio to analyze your data. And also potentially VS Code, where you can now start interacting with and developing your Nextdoor pipelines directly within the platform as well, rather than relying on a laptop or spinning up an EC2 instance separately. The idea will be that you'll be able to do all of this and develop collaboratively within the platform via these interactive environments. And again, we would just be piggybanking off your existing credentials within the platform. And so users can benefit from that. You, as a DevOps or infrastructure type setup team, you wouldn't need to then give access individually to all of these users to be able to do that. We can use your existing credentials to spin up all of this, all of these resources. And automation, lastly. So automation is very important to us. And there are various ways that you can interact with the platform. If you don't want to use the UI, you don't have to. We have an API that you can leverage for your own automation to hook into your existing processes. We also have a command line interface, which so the command line interface is actually built on top of the API, but it just gives you a more user-friendly interface to launch and interact with the pipeline. So anything you can do by clicking buttons in the user interface, you can do via command line. And so bioinformaticians typically love command lines, and I love the CLI, for example. And it just makes it a lot easier for you to automate manual tasks in a nice, easy type of way. When I'm feeling lazy, I can use a user interface as well if I want. So you kind of get a balance of both, depending on how you're feeling. You can even import and export entities like from the CLI, for example. So a compute that I've set up in the platform, it's just code behind the scenes. And so you can import and export that, and you can use that as infrastructure as code to spin these environments up on the fly in a very easy, standardized way. We've also developed a tool called Seqera Kit that sits on top of the CLI. So you have the API at the bottom, you have the CLI built on top of that, and then you have the Seqera Kit built on top of that. And then what that does is essentially it exposes all of the parameters you get from the command line interface in a standard YAML format. And what that means is when you want to set your automation up or have infrastructure as code, you don't need to write JSON. which can be quite fiddly to get right, you can have a simple YAML interface. And for example, on the left there, I can launch a pipeline on the platform by specifying which pipeline I'd like to run, where I'd like to run it, and other parameters and various other things that I'd like to set directly via a simple YAML interface. Again, it just makes it a lot easier and intuitive to run these pipelines. But on top of that, because this package has been written in Python, you can import the package itself in the context of a Python script. And so on the right here, you can see I'm literally just importing that package directly within Python, and I'm able to launch exactly the same pipeline via Python. And so then it allows you to be a bit more dynamic with the way that you set up your automation. You can leverage existing libraries in Python to do more dynamic things depending on your use cases. Okay, so if you're interested, we have a one-on-one demo that we can schedule with you. Get in touch. There's a QR code there. We'd be happy to have a conversation with you and see whether we can help you with how you're using Nextflow internally, and whether something like Seqera platform would fit your particular use case. We have a group demo coming up on the 15th. So again, please feel free to join that. And it's sort of a tour of the platform to give you a better representation live as to exactly what you can do and what some of the buzzwords I mentioned mean. We have a summit coming up in Boston, as I mentioned. We've literally just reopened registrations for that. We have some limited tickets available in Boston at the end of this month. So if you're free and you fancy joining, it's generally quite an awesome conference. It's the first time we're doing it in Boston, actually. And we've got some really good talks and content lined up, especially if you're interested in learning more about Nextflow and potentially the Seqera platform. So thank you for your time, and I'm going to hand it back to Rosie.
Thank you so much, Dr. Patel. That was a very exciting overview of some of your new features that are available. Certainly very comprehensive view for people that need to be able to run, simple or complex workflows. And something I always appreciate is a nice layer on top of that, so I don't have to get into those, super technical details sometimes. So really, really awesome. For our participants, if you had any questions about that, please make sure that you put them in the Q&A now. We'll loop back to them at the end of the webinar. So if something sparked your interest there, or you want to learn a little bit more about one of those features while we have Dr. Patel here, please make sure you put those in there. We'll move along to our next presenter as well, and that is Dr. Brian LaJoie. Dr. LaJoie received his PhD from the University of Massachusetts Medical School, where he worked to develop and pioneer the Hi-C methodology, and he's now a principal scientist with the software and bioinformatics team here at Element Biosciences. At Element, he works with a very diverse set of research and clinical customers with a lot of different asks to build innovative, flexible, and scalable analysis solutions with the accuracy and data profiles of the AVITI system. He works across a very wide range of next generation sequencing applications, a lot of different types of problems to be answered there. So welcome, Dr. LaJoie, and let's learn a little bit about how Element is using all of those great features.
Yeah, thanks, Rosie. Yeah, hopefully you can see and hear me well.
Yes.
Perfect. Yeah. So yeah, so today I'm going to sort of walk you through sort of a use case of how we're combining both the AVITI instrument with the Seqera platform. So starting off with acknowledgements, the team at Element is quite large. Lots of different organizations and functions that are contributing to this work. So I just wanted to sort of thank everybody all at once rather than go through several names here. So just an introduction to the Element technology for those that are not familiar. There's sort of three main pillars or three main things that we think give the AVITI and the technology that we have an advantage. The first of which is a low background surface. So this, of course, is going to help with the signal to noise, which is going to ultimately work to increase quality and decrease error. Second is our innovative rolling circle amplification method, which is quite different from other techniques. This, again, is a way in which we can control and keep error low. And the third is the avidity chemistry, which is sort of the secret sauce to what we think we have here. So there's been a few publications that have gone through the technology. We just had one with Google, and I'm just linking bioRxiv down below. The point here is that you can see our quality scores. So these are read 1 and read 2, different QQ plots showing our predicted quality score and our empirically generated quality scores. So the takeaway here is that the points fall along the diagonal, so our quality scores are accurate, and they reach into the mid Q40s, which is quite high. One of the things that we think differentiates the AVITI and the chemistry that we have is the ability to sequence larger inserts. So traditionally, when you think about a whole genome sequencing library, generally the fragment is maybe 400 to 500 base pairs in length, and you're sequencing the two ends of that fragment. We think, and we've shown, that we can actually sequence inserts that are much larger, so even beyond 1,000 base pairs. And there's lots of benefits to doing that. The primary one is that, at least in human, larger inserts increase read placement. So you can see on the right here, I have a few different panels. On top is a traditional 500 base pair insert library. And you can see the 4 different regions. There's some trouble with placing reads there and being able to make the expected calls that we should be making. And then down below, you see the exact same regions now using 1,000 base per insert library. And you can see that now the reads are being placed, and you're able to actually see the variants come through, and you're able to sort of rescue these difficult regions. So these larger inserts help a lot with these difficult regions. And we know that human has quite a lot of these difficult regions. So the goal here is to sort of demonstrate how we can put this into practice. So just one more example too is we've now created and sequenced and shared 8 total libraries. So these are across HG001 to 004. So we've prepped these at both a 500 base pair insert and 1,000 base pair insert. And you can see the different distributions on that plot there, the 4 different colors at both sizes. Just showing, you can even see that in blue and green, some of these inserts are even larger than 1,200 to 1,300 base pairs in size. So we're able to really begin to push this quite a bit. And the benefit is really, this is a benchmarking exercise. So here we're comparing these standard samples to the NIST truth sets. So we know exactly which variants should exist within these samples. And then we can go ahead and sequence and align and variant call our data and then compare that to these truth sets. And this is counting how many total errors do we end up making. So out of the 3 to 4 million variants that should exist, you can see on left, at 500 base pairs, we have something like 20,000 total errors that remain. And then as we look into the larger and larger inserts, as you move right within this first panel, you can see we can reduce that number of errors by almost 5,000. So the larger inserts are providing a benefit in this benchmarking exercise. And the idea would be that in a clinical setting or in some sort of a research setting, you'd be able to see more into these difficult regions of the genome. And just as you look at the panels on the right, the same benefit is seen across each of the different HG00 samples. So with all that said, so now we want to go ahead and take this and apply this to a problem, a research problem, and then show how we can actually enable the scalable analysis. So everything that Herschel has said, with the Seqera platform, how we can combine that with AVITI and show how to do things at scale. So as a starting point, we're going to apply this to a lupus disease cohort. So the goal here is to really just understand the genetic components of this disease. So this is a 92 sample cohort. We're going to be using PCR-free whole genome sequencing on the AVITI at 2x150, targeting 30x coverage. Preparation-wise, we're looking for greater than 1,000 base pairs in the insert size, and then greater than 90% Q30. These samples have been processed and sequenced by the Element Biosciences service lab, and you can see some of the kits that we're using here with the different kit IDs. And then post alignment and variant calling, tertiary analysis is going to be performed externally by our clinical partner. So that is still in progress at this point, but I'll be able to show you sort of everything from sequencing to secondary analysis today. So just the schematic of sort of what's happening again. So, prep into sequencing, into secondary, and then once you have variants called for each of these samples, there'll be some joint calling, variant annotation, filtering, prioritization, and that'll be driving the tertiary analysis and hopefully some exciting insights into this disease. So if you kind of think about what would be needed to do this, there's a few questions, and I'll go through some of them. So the first is really, where will your run data go? Will you store it locally? Will you store it in the cloud? How will you generate fast queues? Is this something that can be automated? Is this something that you'll have to have, one person or two persons sort of manage? Will you set up file triggers and so on and so forth? Where will the fast queue generation happen? Again, cloud or local, and sort of what tools will be used, right? And then even after you generate FastQ, again, you have to think about where should these FASTQ files go. This is a large amount of data, and this is just a start, right? Some projects are even much larger than this. So where will you store them? And if you decide to use the cloud, some of the key things are, how do you even set up a cloud? How do you set up permissions? How do you set up compute environments? How do you ensure it's scalable? And I guess the big one is really just how much could this potentially cost? And then just keep going through. It's the same for secondary analysis. Where will that happen? What pipeline? So on and so forth. How to QC the results, and then and then the last one is really just how to ensure that the analysis is both reproducible and scalable, right? So that you can sort of have this setup be done once, and then you can apply this to more and more research projects as you go. So this is why we think that the Seqera platform is so great. So we use this internally, and this is sort of what the setup will look like. So for this project, you can see here on the left, I have 7 different AVITI instruments that are being used. So this is just part of our service lab fleet. These are the 7 instruments that have been tagged for this project. So each of these instruments will be streaming data to AWS S3. And then once the run is complete and the data is in S3, the goal would be to run our basis the FastQ software, which will generate FastQ files and perform the demux. And then once FastQ files are complete, then we'd want to run secondary analysis, which is that WGS workflow that you see there. And then once all of that is complete, then you'd proceed on to tertiary analysis. So the first thing is really how are we going to automate or run basis the FastQ automatically? So this is where Element Bio Cloud comes into play here. So if you look at the instrument, Element Bio Cloud is a framework or sort of a system that monitors your AVITI instruments and sort of the fleet of instruments that you may have. And it enables you to configure various different analysis flows. So what an analysis flow is really, you can set a trigger that says, when this run completes, I want to run this analysis. And that analysis could be basis to FastQ. You can also instruct Element Bio Cloud, where you want that analysis to be run and with what options and so on and so forth. So that's exactly what we've done here. Is we've said when these runs complete, we want them to run basis to FastQ on Seqera with the following options and to deposit the FastQs back into this bucket on S3. So as operators in the lab go and set up these runs, nothing needs to be done in terms of starting the analysis. It's all sort of set up on the front end, and then Element Bio Cloud will manage that and orchestrate the workflow launches with Seqera to be able to get the results that we want. So one other complication with this analysis is that some of the pooling that we've set up for these samples, often we will have to combine FastQs across runs as input into whole genome sequencing. You can kind of see that in the diagram here. And just to get into some of the details around what is actually going to be happening within this whole genome sequencing workflow, you can see some of the tools we're planning to use on the right. So, alignment is going to be powered by Sention, same with small variant calling. We're also going to be leveraging Google Deep Variant. and then a few different callers to be able to get at CNVs and SVs and SVRs, and then of course a wide range of metrics. So with all of that set up, this is sort of how things look when we actually went ahead and began the sequencing. So on the top left here, you can see that this is sort of a picture of what the Element Bio Cloud looks like. And the primary trigger is sort of this, this graphic that shows run status and ultimate run completion. So when the run completes, basically the data is going to be deposited into a S3 location of our choice. And this is sort of what a run looks like. So you can see a run name, various different JSONs, and a run manifest CSV, along with the actual run data. So Element Bio Cloud receives that trigger. It realizes the run is completed. It then talks to Seqera platform through various different APIs and kicks off the launch of a basis to FASTQ workflow. So this is a picture of what the Seqera platform UI looks like. This is that basis to FASTQ workflow. So you can see there's just a single task there because this is a quite simple workflow. It's just running one tool, right, to generate FASTQs and perform the demux. So that's what we end up seeing within the Seqera platform when the automation triggers. And then, of course, this is just repeated for each of the different runs as they're sequenced, over the course of a week or two. So we end up with 59 total workflows that have been launched on Seqera. And when these all complete, at this point, we have all of the FASTQs that we're going to need for this project in our desired S3 location. So one thing that's really great about Seqera and the various different APIs and metrics that they provide is that we can really get into the nitty-gritty details of how much things cost and how long they take to run and optimize in many different facets. So you can see here, this is the actual incurred cost of running basis to FASTQ for each one of these runs. So each bar is just a given run, right? So there's 59 runs, 59 executions of basis to FASTQ. So on top we have cost, and on the bottom we have the actual time that was elapsed to sort of run that workflow. So you can see that the cost and the timing is quite reasonable. In total we spent $20 to generate FASTQs across these 59 runs, which means it's something like 30 cents per run to do this. Average runtime was 19 minutes or so. And you can see some of the actual compute that we are using for this. And this is all orchestrated through Seqera. So AWS Batch, we're using Spot EC2s, and we're using a specific M512X instance that sort of has a higher amount of CPU and memory just to enable sort of rapid execution. But because we're using Spot, the cost is quite controlled. So we can also, now that we've actually generated and ran this workflow across these runs, we can look at some of the metrics. So you can see the actual base quality across the 59 runs. It's quite robust and reproducible, so it's always above 40. So you can see most of the ranges in the 42 to 43 quality score range. And you can look at that same quality score by cycle, and you can see that a high quality score is maintained across almost all 300 of the cycles, always above Q40. Same with percent Q30, so we met our spec. All of them pass our QC, and same when you look across cycle. Quality is quite good across the entire, all cycles and across all runs, which is great to see. So, with all of the FASTQs generated, now we'd want to move on to running whole genome sequencing. So this is sort of what the output of a basis to FASTQ run looks like. So you have this QC HTML report, which is a great, interactive report, similar to multi-QC in many ways, that you can use to dive into the details of your run, various different metrics and JSONs and whatnot. And then you have these sample directories where for each sample that was in your manifest, so here's some PyX samples and whatnot, and then our sample of interest right here. So with all of these in S3, again, there's a little magic with Element Bio Cloud and Seqera to sort of orchestrate the launching of WGS. So it's sort of looking at which FASTQs we should be using as input, and then providing those to Seqera through the API to basically launch our whole genome sequencing workflow. And here you can see now we have a lot more than just one task because there's multiple steps within WGS. And of course, this is repeated per sample. And then here's sort of what things look like as every sample is launched. So in total, there's 92 launches here running whole genome sequencing. These are quite resource intensive, but of course, because we're on the cloud and we have the power of Scale and Seqera, we're able to run all of these or even many more concurrently. So the cloud is quite infinitely scalable. So again, with all of the metrics that Seqera provides, now we can go ahead and look at cost. So this is sort of a cost of, I'm just showing two of the steps. I think there's something like 40 steps within our workflow, but the two primary ones are alignment and variant calling, right? So you can see the cost of alignment, quite reasonable, right? Same with the time, quite reasonable as well. So this is something like 50 cents. And then the time to align is maybe less than 30 minutes or so. And then variant calling as well, 50 to 60 cents to do the variant calling. And you can do that in, let's say less than an hour, the actual variant calling. And of course, these are, and here's some of the actual compute that we're using. So we've chosen to align with a much bigger instance, where we use a smaller one for variant calling. Of course, if we wanted this to be faster, we could have just simply allocated or sort of requested a larger instance, and we probably could have cut that time in half. So, here's another great plot that just shows, again, cost and time across every step within our workflow. So this was something that was very easy to pull out of the Seqera API and metrics that they provide. So you can see, probably the most expensive step is the alignment, as you would expect. That's probably the most resource intensive one. There's a few other that are sort of a little bit higher in cost. But this really gives us a lot of power to sort of look at our workflow and optimize and decide, are we using the right instance class? Are we using the right resources? Are we wasting resources? So we can use this data and really refine each individual step within the workflow to optimize for whatever it is we want to optimize for, whether that's cost or speed or whatnot. So you can see in total to run those 92 samples through our whole genome sequencing workflow, we spent $422, which again was provided by Seqera. And if you really work that out, it's $4.50 per sample to basically run the analysis to go from FASTQ to VCF for each one of these human samples. And again, we're using batch, spot pricing yet again, and then various different instance classes or instances from this C5, which is a CPU class. So showing some of the metrics so now that we've run WGS, we can look at many metrics, right? So here is what the coverage looks like. So we were targeting at least 30x coverage, and you can see for each sample, it's a bit variable depending on sort of how many runs that sample was sequenced on or how the pooling worked out, but everything sort of looks as we would expect, and all of them have the required coverage that we'd be looking for. Looking at insert size, so if you recall, again, we were aiming to sort of prepare these libraries to have greater than 1,000 base pairs in insert size, and you can see 1,000 base pairs is right here. So all of these are quite large. I think the mean is 1,200 base pairs, 1,200 base pair mean size, so quite large. But then the distribution is quite broad too. So there's quite a lot of sizes even beyond 1,500 base pairs or so. So quite exciting to see that. And of course, seeing that works well at 92 samples is nice to see. Same with quality. So now we're looking at quality by cycle per sample. Again, you can see that the high quality score is maintained across, all 300 cycles, and it's almost above Q40 for sort of every cycle. So that's great to see too. That means we're going to have, very low error and high quality as we get into the variant calling and analysis. And then, one more metric too is duplicates. So we've shown before that the duplicates are generally very low on the AVITI platform, maybe compared to other competing instruments. So you can see here the total duplicate rate is let's say less than 1% across, or right around 1% for all samples. And all that really means is that there's going to be more usable data on an AVITI platform and maybe a different platform that is more prone to having duplicates or optical duplicates. So, at this point, whole genome sequencing is run. We have all of these various different BAMs and VCFs for each of the 92 samples, and this is what's still in flight. But at this point, the data would be going into sort of a joint calling and a clinical interpretation platform to drive, hopefully, some very exciting scientific insights. So I don't have anything to share on this just yet. This is something that our partner will be sort of leading, but we hope very soon to have a conclusion to the story in a publication to really describe what it is that they have found. So this is just an example of one way in which you can leverage both AVITI and Seqera to drive a research question at scale. We focus on whole genome sequencing, but the great thing about Seqera and Nextflow and NF-Core is that there's all of these standard pipelines that are available. So this could have been an RNA-seq study. It could have been single cell. It could have been, we could have even used this rare disease pipeline, and that's something that we're looking at as well, is whether or not we can leverage this instead of the WGS pipeline that I showed you today. So there's lots of power here, especially with NF-Core. And just two more things I'll show you, and Herschel did mention this. So this is multi-QC. So we have a pull request in now to add bases the FASTQ support to multi-QC. So just as you can use multi-QC to look at metrics across whatever tools you're using now, we're going to be enabling bases the FASTQ metrics. So here's the exact 59 runs that I showed today, running through the current branch that we have of multi-QC with bases the FASTQ support. So you can see, all of the standard metrics that you'd expect to see are there. So we hope that this is released very soon and folks can start using this. And then the last thing is really just more future looking into Element BioCloud and what that looks like and what that's going to mean. So the focus is really on automation and making it so that setting up various different integrations is very easy. Being able to browse files through various different connected accounts, which could be storage or various different analysis platforms, and then tracking and visualization of various different analysis flows. So making sure that you're able to set up an analysis flow and then monitor the execution of that analysis flow that's happening on some external platform. Yeah. So I'll stop there and hand it back to Rosie.
Thank you so much, Dr. LaJoie. That was a very interesting presentation walking through a complex case study of a lot of data. I would like to invite everyone to use that Q&A session if you have any questions right now, and we can have a discussion here about some of the great benefits of the Seqera platform and how it works with AVITI, and also answer any of the questions that you guys have. So I'll get it started. With a question that I get a lot when I'm on calls with people. And they're kind of setting up and looking at, what analysis they want to do. And I get the question of, how do I set this pipeline up? I know I need to do 2, 3, 4 steps. Do I put it all in one EC2 instance and make one big one? Do I try and split it up? Do I use some triggers, some flows, etc.? I don't know what to do. So Herschel and Brian, if you could maybe comment on what's the best way for a user to dive into that problem with Seqera and maybe picking out the steps of their pipeline? How does someone take that question and really put it together?
So I can go first and maybe pass on to Brian afterwards for his thoughts. So I guess the answer will be, it depends a lot on the context, what we're talking about, what type of pipeline, what infrastructure you've got available at your disposal to attempt to automate stuff. So Seqera platform exclusively runs Nextflow pipelines. So before you start writing a pipeline, you'd need to ideally have some background knowledge of Nextflow and what it can do and that sort of thing. There are tons of training courses, both recorded and also in the future that are being run by the community. And at Seqera, we also run these training courses for our customers as well. So first of all, I guess it's learning Nextflow to some sort of intermediate level of knowledge. And then really it's breaking the problem down in terms of what you're trying to achieve. And in some instances, it might make sense to have a very small pipeline like the basis to FastQ one that Brian showed, because then you want to do something very specific and you want an endpoint there before you start then deciding whether you want to run an RNA-seq pipeline or a demultiplexing pipeline, or sorry, an RNA-seq or a single cell or a rare disease pipeline. And so that kind of makes sense as a separate module or pipeline itself. And then you would have more complex sort of pipelines downstream that are performing a series of steps to get you to another endpoint that you can kick the can down the road to something else afterwards to take a bit further on. So without being too definitive, I think the answer is it just depends on what you're trying to do. And you just need to do what makes sense for you in terms of how you want to analyze that data and compartmentalize what you can and make it as modular as possible.
Yeah, and I'll just add that most pipelines are quite complex, and there's lots of different steps, and many times there'll be one step that is quite resource intensive, where you want specific set of CPUs or memory, and then maybe you have to go through some metric calculation that only supports using one CPU. So you don't want to be on a giant instance with 96 CPUs and then be spending that money, while you wait for a couple hours for metrics to calculate. So that's why it's so great about using Nextflow. Each task can have its own Docker image, its own container, its own set of resources. They could be GPU or CPU, and you can really fine tune all of that and make your workflows both efficient for time and cost.
Great. We've also spoken a lot about doing this in the cloud here too. For some of those people that have maybe the hybrid resources, local and cloud, or working locally, is it possible to mix and match those with the Seqera platform?
It is, but it may not necessarily be the best way to go in general, because if you think about that, what you're essentially doing is, say, for example, you've got a HPC in your organization and you also have access to a cloud account and you want to do some form of compute on your HPC and then delegate the rest to your cloud account. The main limitation with that is the fact that you need to move all of this data around. So you need to, you may want to do some processing, and it's possible you can do it with Nextflow, where you can have process level directives or executives that say, for this particular process, run it on my HPC. For the rest of the processes, run them on AWS Batch, for example. So it's possible, but I think you need a very big cable to make sure that you're able to shuttle all of that data between your local and cloud environment in order to make that actually worthwhile doing, to be honest.
Okay, great. Very interesting. And then Brian, maybe you could speak to us a little bit more about that multi-QC. What am I going to learn from using multi-QC with basis to FastQ? What metric can I get out of that? And maybe when will that be available too?
Yeah, so the HTML report that I mentioned briefly, so we do provide a report that has all of the standard metrics per run. So, quality, demux stats, indices that may have not been assigned within your run, percent Q30, percent Q40, so on and so forth, right? Multi-QC comes in because now instead of just looking at that report per run, you can say, let's aggregate the metrics across these 59 runs or these, 1,000 runs. And then you can look at all of those runs together in multiple different plots. So you can look for outliers, or you can do all sorts of neat aggregate analysis using multi-QC. So yeah, it's quite powerful. And of course, multi-QC already supports all of the other tools that folks commonly use. So now you can sort of have one view into both FASTQ generation, demux, and then whatever secondary you happen to be running. So it's quite great. In terms of the timeline, we have the code in now. We just need to work through a couple changes to get things approved and playing nicely with the existing multi-QC code, but we're close. So I'd expect a couple weeks at most, and then it should be released and folks can use it.
Something very exciting to look forward to. I'd like to thank both of our speakers today, Dr. Patel and Dr. LaJoy, for sharing their expertise. viewpoints and walking us through something that can be very helpful for a wide variety of different applications. Really emphasizing that AVITI can't always fit for you. There's a lot of different applications out there. Here's two great ways to kind of get your data and process your data together with Element and Seqera platform. Just in closing, a couple more announcements before we wrap up. First of all, just a reminder for people about that AVITI for All Accelerator Grant. You can get a link right on our homepage, elementbiosciences.com. Applications are open, so go put an application in, check it out. If you have any questions, let us know. This webinar was recorded. I will have it available for download in the next several days. Keep an eye out on your inbox for a follow-up email with the link to the recording. And last but not least, this is 2 of 3 in our webinar series, AVITI, Always the Right Fit. You can register for our next webinar, which is going to be on Tuesday, December 5th. Registration information is going to be on our website. It's going to be on our socials. So keep an eye out for that next step in this webinar series as well. Thank you again, Dr. Patel and Dr. LaJoy, and thank you everyone for joining us today on this webinar. Take care and have a great day.